While most research conducted at Brooke Army Medical Center involves human subjects, Department of Clinical Investigations is able to support laboratory and cadaver research. Here to present on those topics are Captain Daniel Dees and Mr. Eric Steele. Captain Dees, the laboratory manager at Brooke Army Medical Center, is a board-certified medical laboratory scientist with 13 years of experience in the clinical laboratory.
Good afternoon, everyone, and welcome to the final presentation of CERT 2021. I am Captain Daniel Dees, the laboratory manager here at DCI, and I am joined today by Mr. Eric Steele, DCI's resident molecular biologist and research consultant. Now that you've had a full day's worth of valuable information on the complex processes associated with military research, we are here to talk to you about the capabilities BAMC DCI possesses that can help you reach your research goals. I'm going to get us started by a broad overview of DCI laboratory capabilities, our oversight of cadaver research, as well as a quick plug for animal research at the ISR here at BAMC. So let's go ahead and get started. First off, with the standard disclaimer, just letting you know the views expressed in this presentation are ours and not any of the entities listed here on the slide. And we'll jump right into the automation that we possess here. DCI has automated sample preparation available to extract, purify, and amplify targets of interest. Automation helps decrease the human error factor and allows us to minimize hands-on time to decrease the wait for what we'll all really want to see, results. On this slide, you see a few of our analyzers, starting to the left with the Chi-Cube for DNA RNA extraction, next the Chi-Agility for medium throughput pipetting and sample prep, and finally the Chi-Symphony, which combines the process into one analyzer for larger volume projects. So as you can see, DCI laboratory workflows are fully scalable to meet the investigator's needs. Aside from the basic molecular biology capabilities, DCI also houses a state-of-the-art pyrosequencing analyzers capable of detecting epigenetic modifications that regulate gene expression. Last year, DCI became the first laboratory to use epigenetic profile data acquired from the PyroMark Q48 platforms you see here on the slide to publish in a peer-reviewed journal. Among the newest addition to the DCI laboratory capabilities are the FlexMap 3D, capable of simultaneously measuring up to 500 genes or proteins from a very small amount. One of the more promising applications of this analyzer, which Mr. Steele will discuss later, involves detections and quantifications of cancer biomarkers. And we're actually reaching out currently to different interested parties to develop more cancer biomarker projects. One of the more interesting analyzers we have here at DCI is the IncuSight Zoom. It allows us to view, record cell culture growth and manipulation in real time by recording continuously and condensing those videos into time-lapse videos, like the ones you see here on the right continuously playing. This is actually an apoptosis assay where you can see the red intact cells are bursting, releasing the green fluorescence. So you can see in real time how your treatments are having effects on the cells listed. A few of other applications with the Zoom are immune cell killing, antibody internalization, phagocytosis, and live cell immunochemistry. So now I'll go a little bit into the cadaver research here at DCI. We're responsible for the oversight, the regulatory guidance of cadavers. So let's first talk about a little bit what a cadaver is. So we know a cadaver is a deceased person, but also it's any portion of the deceased person. That includes organs, tissues, eyes, bones, arteries, or any other specimen obtained from an individual after death. So this does not include things that were taken off of a live individual, such as organ, like a live kidney transplant. Some of the ways that we use the cadavers, one of the more regulated uses is sensitive uses. So these include exposures of cadavers to impacts, blasts, ballistics testing, crash testing, other destructive forces, because we want to be sure that we're using our cadavers in a very respectful and scientific way. So now that we know what a cadaver is and what are the sensitive uses for it, we can go over the flow chart here on how to actually submit cadaver research to DCI here at BAMC. So the PI or program director submits a detailed plan to DCI. DCI reviews the protocols for all the legalese that needs to be there. Once a determination has been made, whether the protocol meets all the applicable requirements, it either gets submitted back to the PI, our program director, for corrections, or it continues on to the next question. Is it going to be included in any of those sensitive uses that we described in the previous slide? If the answer is no, the activities may commence. If the answer is yes, then DCI submits the protocol all the way up to Stratcom to headquarters at MRMC for review. So if no objectives were found within 15 days, a 15 business day period, the activities may commence. If there's any issues, it gets kicked all the way back to restart. So as you can see, it's kind of this makes it looks very easy here, but it is a very complex, time-consuming procedure. So, but that's what we're here for. We're here to assist in any way possible. So please, if you're ever interested in any type of cadaver research, reach out to us and we can get you started on the right path. And earlier today, we heard from Colonel Stackle on the ISR, so I won't beat this one into the ground, but I did want to give a little bit of plug because we do give our support to the ISR and some of the protocols that are done there. One of the ongoing protocols is a microvascular training you see occurring over on the side. So we provide veterinary technicians that we have here on staff to go down and actually assist in the protocols as needed. Similar to the last slide, the kind of the flow chart for submitting animal research is on the left. This is extremely simplified, but this is all on the intranet page. The DCI intranet page, if you click on animal research, this will lead you step by step, and it even provides the templates that you need if you're interested in animal research. And with that, that's the end of my portion of the presentation. So I'm going to pass it now over to Mr. Eric Steele for the second half of the presentation. Thank you all.
While most research conducted at Brooke Army Medical Center involves human subjects, Department of Clinical Investigations is able to support laboratory and cadaver research. Mr. Steele is a microbiologist with experience in the collection and analysis of clinical isolates for multiple drug resistance as well as an extensive molecular analysis experience.
Hello, my name is Eric Steele, and I would like to introduce you to the Department of Clinical Investigations Research Consultation Section. DCI has a variety of automated sample preparation platforms to help you streamline your process and increase consistency. Some of the studies that have utilized our resources in the past have been an environmental metagenomic analysis of the various duty forms worn in the emergency department to determine the potential for pathogenic bacteria being present on the clothing that could come in contact with patients, and if there was a discernible difference across the types of uniforms worn. Another protocol was in collaboration with Army Public Health Command to determine the risk of contracting tick-borne relapsing fever by optimizing extraction and detection assays for the Borrelia species responsible for the disease carried by soft ticks that are present in the Joint Base San Antonio training area. As Captain Dees highlighted, we have dedicated pyrosequencing instruments that are the current industry standard, ideal for epigenetic modeling studies. This technology is currently being used to study the impact of adversity on epigenetic markers and how that impacts the outcome of patients that have suffered mild traumatic brain injury. The lab has recently acquired the ultimate in high-throughput multiplexed protein quantification for the evaluation of biomarkers for disease process and treatment efficacy. One study evaluated the limits of detection for thrombospondin 1 and cathepsin D that are being investigated as potential biomarkers for the detection of prostate cancer. Another study evaluated biomarkers associated with inflammation to evaluate if treatment of lower back pain could be quantified. A recent protocol studied high-level disinfection of surgical instruments due to the increased emphasis being placed on prolonged field care that might be required by military providers in far-forward, austere locations. Multiple solutions were tested to try to find the best candidates based on how long it took to effectively eliminate bacterial contamination. There is currently an investigation of prenatal surveillance samples taken from expectant mothers determining the presence of GBS colonization with the intent to elucidate the endemic Group B Streptococcus agalactiae strains in the local military population. Whole genome sequencing will be performed on the culture bacteria to determine the prevalence of hypervirulence and antibiotic resistance genes and their relationship to the pathology of disease and disease outcomes. Remember, we have a dedicated staff of laboratory professionals that can help you with study design, platform choice, and training to get you started on your research project. Please feel free to contact us. Thank you for your time, and have a great day.